Oilseed rape is very important for the UK agriculture because it's the third most important crop. Its uses are in food production as well as the protein meal used for animal feed.
The virus, turnip yellows virus, is very important in oilseed rape. We've found crops where there's been 100% infected, and it can reduce the yield of these oilseed rape crops by in excess of 20, 30 or perhaps even 40%. It's also very important in vegetable brassicas where we've seen yield reductions of up to 60%.
So the economic impact of a disease like this can be as high as £180 million for the UK, but it does vary between 67 million and 180 million. The incidence of turnip yellows in the UK is estimated about 60% the acreage of oilseed rape fields, but in Europe it's even higher, and at those high levels of incidence we're going to see very large losses in yield. We see very high incidences of turnip yellows virus in both the UK and Europe because it's readily transmitted by the aphid Mysis persky, obviously, which is one of the largest crop threats pests that we see. And it has a high efficiency, above 90% of transmission, as well as we found around 80% of Mysis persky actually carry turnip yellows virus. And once Mysis persky has acquired the virus, it's able to transmit it for the remainder of its life. Not only are aphids very readily able to spread the virus, but many weeds are reservoirs to the virus. So not only do we find it in crops, but we find it in species like dandelion or nettles that you might have all over the country. So they are a source from where these aphids can feed and then transmit the virus into crops. One of the struggles farmers have to actually identify their crops being infected with turnip yellows is that it has very muted symptoms. So it's very difficult to tell what's just an aging plant with what's actually an infected plant. Previously and existing are pesticides such as neonicotinoids, which unfortunately have now been banned, which were an effective means of controlling the virus by controlling the aphid itself. But with them being banned and the ineffectiveness of other pesticides, the only sort of way to control turnip yellows virus is by having natural plant resistance. Mysis persky is gaining resistance to all the major chemicals used to control them, including now neonicotinoids, and with the lack of chemistry and the ineffectiveness of pesticides, this is becoming harder and harder to control turnip yellows virus in the field.
So our approach to controlling the virus is to look at natural plant resistance. So we've identified a number of sources of natural plant resistance in different brassica types, and our aim is to get these into commercial crops.
To combat turnip yellows, we have obviously sourced three plant lines, Brassica napus, Brassica oilacea and Brassica rapa, with different sources of resistance in each line. And the aim is to breed these into commercial lines to produce resistant varieties. To ensure that all our resistances found in our three lines were going to be durable, we tested each line vigorously against the different genetic groups we found of turnip yellows virus in Europe, and each line turned out to be resistant to all three genetic groups. So we're confident that this will be a durable resistance to be able to use in the field. We've managed to produce major QTLs for each of the three sources of resistance, as well as linkage maps. So this will help us identify the gene of resistance, as well as produce better markers to follow the resistance for introgression into commercial varieties. We've also managed to produce a resynthesized Brassica napus, which has introduced the resistant source from Brassica rapa and Brassica oilacea that we produced, and this resynthesized napus has gone on to double its chromosome, and we've started to cross it with commercial varieties and self itself, so it is fertile. So this is a big step at producing a new resistant oilseed rape line to put into commercial varieties.
So we have found resistance in oilseed rape, but we've also found more extreme sources of resistance in Brassica oilacea lines and also Brassica rapa lines. And what we've been able to do is actually to make crosses between those two species and resynthesize Brassica napus or oilseed rape from these two very resistant lines. We've provisionally mapped the QTLs that are involved in these resistances in Brassica napus and also oilacea and Brassica rapa, but we need some future research to try and get some markers that are closer to these QTLs so that we've got reliable markers that will help with the integration of these resistances into commercial oilseed rape lines. So we've got the materials, we've got the expertise. What we're looking for now is for some commercial involvement in this project so that we can get some funding from the BBSRC to take the research forward and drive this through to producing commercial varieties. For further information, you can look at our website, and also there'll be an email address there where you can contact us.